This is microbial phylogenomics. I'm Jonathan Eisen. I'm a professor in a bunch of different places. My lab is in the Genome Center, as many people here know. The Genome Center is sort of a cross-departmental association of faculty, and my main appointment is in evolution and ecology, which is why that's where this course is being offered, although the course does have an evolution and ecology focus. And I also have an appointment in the medical school in medical microbiology and immunology. What this course is largely about is all the things that I work on. I'm not going to talk about my own work much, but this is the topic of what I work on. You'll hear lots more about that during the course. For those of you who've taken BIS 2C with me, I like to remind people where we've been and where we're going when we have a class. So we haven't been anywhere before, although hopefully everyone here has taken some form of an intro bio series, because we're not going to cover, we're going to go into a lot of detail on molecular biology and DNA and genomics and things. We're not going to do a lot of the background. Today we're going to focus on just sort of an introduction to the course, an overview of the course, some of the details on what's going to be expected in the course and what I hope people will learn in the course. And then the next lecture we're going to talk about what I call the evolution of DNA sequencing. So and as you will see throughout the course, what we're going to do in each topic. So there really are 5 major topics in this course. And for each topic we're going to do the history, a bit of the history of the field, and then where we are now in the field. And for some of these topics, that will be compressed, and in other topics, it will be expanded greatly, such that we will spend a bunch of time not just on where we are now, but on case studies of particular papers or particular scientific studies that have been on those topics of where we are now. Basically for today, again, we're just going to go through these course details and talk about what I call the 4 eras of DNA sequencing, and we'll come back to that. And we probably won't get to the little bit on phylogeny, but if we have extra time, I thought I would review some phylogenetic tree stuff for people if you have forgotten. So again, these are the 5 topics in the course. So DNA sequence-based studies of microbial diversity is the overview. Everything we're covering in the course relates to that, and we're going to spend a little bit of time on just, one lecture, basically, one class on DNA sequencing itself. You don't really need to understand all of the details, but it's important to see the landscape of what has happened in the past and where we are now, because everything that people are doing related to using DNA sequence data to study microbes has been completely blown out of the water and revolutionized by new technological advances in the ability to read the string of letters in DNA sequences from different organisms. And then we're going to go through what I call these 4 eras, but they're really overlapping eras in a sense. And we're going to spend a few lectures on each of these topics. And again, for each of them, we're going to do basically 1 lecture with a couple of papers to read on the history of the field, or maybe 2 lectures that's going to go through like how this whole area started, and then 1 or 2 or more classes on what we know now related to that topic, and then 1 or 2 lectures on a case study of some recent paper that covered this topic. And these 4 topics are, and I'll go through this in more detail, the tree of life, that is evolutionary relationships among organisms, environmental studies of microbes, looking at this one gene, the ribosomal RNA genes, where people were able to look at microbes without ever growing them in the lab. And then genome sequencing, reading the entire sequence of the genomes of organisms that you could work with in the lab. And then something called metagenomics, which is genome sequencing from the environment. So basically the way this works is the first era started in the 1970s. People could grow microbes in the lab, And to figure out the evolutionary relationships among them and the tree of life, people used sequencing. That was how the whole field got launched. Now that keeps going today. People are still studying the relationships among microbes. But then in the 1980s, people said, well, that's great for organisms that we can work with in the lab, but what about organisms that are out in the environment that we either can't grow in the lab or don't want to grow in the lab, or it's difficult to work with in the lab? And what people did in the 1980s was read small pieces of one gene from these organisms, and that was used to basically do the equivalent of what people have been doing with plants and animals with binoculars and other types of techniques to study microbial diversity in the environment. And then in the 1980s, the first complete genome sequence of any organism was determined, and this was of a bacterium. And after that, there's been this sort of revolution of organisms that you work with in the lab. Rather than just reading one gene from them, you can actually read the entire string of letters in their entire genome sequence and learn a lot about their evolution, their biology, their diversity, et cetera. And just as with the tree of life, working with cultured organisms wasn't enough, people then said, well, as DNA sequencing got cheaper and faster and easier, let's go back to the environment, and rather than just reading the sequence of one gene from the environment, let's read the sequence of the entire genomes of organisms from the environment. And this is, of course, not everything that people do for studying microbial diversity, but because DNA sequencing has gotten so cheap and so easy, this is now a fundamental tool to almost every single study of microbial diversity in any ecosystem anywhere, some type of DNA sequence-based study. And so this is basically, that's the course here. And again, for each of these, we will do this sort of layered history for each class. We're going to have usually some primary research publication and maybe a review paper related to the topic. You're going to be expected to read them before the class. Starting next week, there will be weekly assignments related to the reading, simple, short assignments, but just to make sure you're actually doing something. And then we will discuss in class those papers and other things related to those topics. Not sure how to introduce this, but there's a large movement of which I am part of to try and make scientific literature freely available to the community, and not just freely available, but what's called openly available, which means that you can do anything you want with that publication. You can take a figure from that paper and post it on a website, or print it out on the street corner and sell it if you want to, if you can con someone into buying it. Whenever possible, we're going to use open material for this course, which will allow me and anyone else to then take that material, and I will be blogging about this course as we do it to try and engage a broader community, and it's convenient and better generally if you want to post something publicly to have material be freely and openly available. And depending on the topic, some of this will work and some of this won't. We will also include in the weekly assignments, it may not be a paper, it may be a blog post from somewhere, it may be a new news story when we talk about fecal transplants in humans. There was actually a long story in the Washington Post about it today, or other type of media. What you should learn from this course, I mean, hopefully many things, but this is sort of the four main areas of things that you should expect to get out of this course. The history of the field, that is the history of sequence-based studies of microbial diversity. Current practice, so not just the history, but what are people doing now? So these are basically approaches. How do people study microbial diversity? And that should also teach you about science in general. How do people approach scientific research topics? We're also going to learn about what we actually know, so at least current models for facts about microbes and microbial diversity and various ecosystems, depending on the case studies that we do. And one really important thing, probably what I consider the most important thing, is we're going to get practice and I will talk about general approaches to how you read and analyze a scientific paper. This is, a fundamentally important thing, not just for doing scientific research, but for interpreting any other scientific claim that is out there. So we're gonna, take even, classic Nobel Prize winning scientific studies and say what's stupid about them, or what's good about them, and so on. The grading model for this course is basically a fifth assigned to each of these categories. You're going to be expected to be here. Probably might have some type of class participation requirement, like taking turns presenting something about papers. Nothing particularly onerous. There will be these weekly assignments starting next week that are basically going to be different each week, but relatively straightforward, making sure that you've read the papers or read the blog posts or understand them at some level. We're going to have a short take-home midterm, and then a final exam. And in addition, probably the sort of key part of this course is you're going to be expected to do a presentation to the class at the end of the class. And that presentation is basically going to be you independently selecting one or two papers on a topic related to something that we're doing in the course. Review the paper, write up a brief summary. We'll give you more detail on this later. Post your assessment, your summary to the course blog, and then engage with the other students. So you're going to be, part of your grade in this assignment will be that you read the other people's presentations about their projects and ask some questions about them, as well as responding to your own. Okay, so I will have office hours. I've been out sick, so I haven't figured out exactly when my office hours are going to be. And Holly Gans is going to be the real brains behind the course, teaching assistant. She works in my lab as a staff scientist. She's going to be helping out with just general things related to the course, although with a small group, it's not like it's going to be complicated. There's a smart site set up. I haven't populated it with tons of information, but we have started to post PDFs of the papers that we will be reading there under resources. There's a folder under resources for papers. I will post the slides, which I did an hour or so ago, under resources, and we will start posting a variety of other things on that site. Does everybody here use Smart Site? Everybody's signed in through. There's also, for this, as I said, I'm going to actually be blogging and posting information about this course publicly. And I'm going to be doing this through a site called MicrobeNet, which is a project that we run in my lab for communicating information about microbial ecology, basically, although it's focused on microbial ecology of buildings and the built environment. And that's the website where we're going to be doing it. I'm hoping that almost everything will be cross-posted to Smart Site, but some things don't work very well within Smart Site, so I may end up pointing you to this site for a few YouTube embedded videos or things like that. So before we get into the detail, I thought it would be good if we can go around and you can just say your name, what year you are, what your sort of interests are, if you have them, and 2 seconds, 2 sentences, 20 minutes if you want. So, all right, I'm going to start with you, Holly.
Hi, I'm Holly Gans, and I'm just here to help out. And I'm working on microbes in buildings for animals live as part of this microbe.net project.
Microbes in what?
In animal shelters and zoos.
Meaning animals that are in shelters waiting foster homes, or?
Right, like Yolo County shelter where people who can't keep their animals.
You're talking about beneficial microbes or about?
Everything.
Everything, yeah.
Everything. Really? That's great.
Yeah, we just talked about it on the microbe.net blog about a paper where they looked at dust in houses with dogs and that comes from being an infant or maybe prenatal in an environment with animals.
So they are media for beneficial familial microbes.
Yeah, we share a lot of microbes with our pets. We should have. But of course, a lot of deaths in your house isn't necessarily good for animals. So it's complicated.
Yeah.
My name is Elisa. I'm a fourth year. My major is evolutionary ecology, and I enjoy my major and baking.
Hello, my name is Kelsey, and I'm a first year genetics grad student. And I'm interested in genomics, like comparative and functional genetics.
My name is Patricia. I'm a senior. I'm an anthropology major, and just really interested in genetics, particularly in
Hi, my name is Sandhya, and I'm a fourth year genetics major, and I'm really interested in all the genetic diseases and all that.
I'm Jennifer. I'm a third year. I'm a microbiology major, and I'm actually really interested in what this class teaches for future research.
Hi, I'm Shakia. I'm a fourth year, but I'm switching into genetics because I'm crazy. And I'm interested mostly in like how the genomic sequence of viruses and how they change. Yeah.
I was an East Asian Studies major for 2 years before switching. So switching's good.
My name is Mark. I'm a carpenter, and I was reading a Nature magazine article on how we were primarily bacterial cells rather than animal cells. And I was reading up on some of the Fenter group's work, and I decided I'd quit carpentry, building microbial.
Yeah, we could use you.
And I talked them into letting me in here because I'm just wildly enthusiastic about it. I'm actually a student.
My name is also Mark. I'm a fourth year biosci emphasis in evolution ecology, and I'm interested in genetics and evolution broadly.
I'm Grant. A fourth year is my last quarter, and I'm an evolution ecology major. I just haven't really taken any microbial oriented courses, so I figured I would load them all up this quarter. I'm taking cell biology this class and physiology course.
My name is Roxanne Baker, and I'm a first year graduate student in the geology department, and I'm interested in this course as or possibly helping me with my research.
And my name is Joanna. I'm from Brazil, and my major in Brazil is agronomic engineering. And I really like this part of genetic area.
My name's Jason. I'm a fourth year genetics major, and I'm interested in bioinformatics approaches towards immunology.
I'm Hannah. I'm a fourth year biochemistry major, but I guess I'm interested in a lot of things, primarily, I guess, microbial ecology and related fields.
I'm Alex. I'm a third year microbiology.
You had to think about that?
Yeah. Third year microbiology major and just took on an EVE minor. We'll see how that goes. I'm interested in microbial ecology and environmentalism, maybe? How microbes might relate to that? Maybe?
It's good. I change every 6 months, so somehow it's always about microbes, but what it is about microbes always changes. Yeah.
What's your medical school appointment about? What is it that you specialize in over at the medical school?
There's a presumption in your question that I know anything about medicine. The medical microbiology and immunology department here, primary focus of most of the faculty in that department is on mucosal immunity, and either the host side of mucosal immunity or the microbial side or the interaction between the host and microbial communities. So surprisingly, I fit in incredibly well in that department because at least half of the department focuses on the microbial diversity of some model, either human or mouse or monkey microbial gut community. So that I don't do any it's not a clinical department. It's a basic science department. So all of the departments in the medical school that are clinically focused moved to Sacramento a few years ago to be associated with the hospital and the new expanding medical research and education facility there. The departments that are much more heavy on basic science stayed here on campus. So most of the people in the department don't do any clinical work. A few of the pathology, the people who study pathogens, a few of them do clinical work at the hospital, but most are just basic science. And we'll go through this in a little more detail. What I'm generally interested in, which we probably won't talk about a lot, is how organisms evolve and invent new functions, the origin of new functions. And for 10 years or so of my career, I studied this by looking at an organism itself and how its genome changed and allowed for it to invent new functions, either by inventing a new gene or taking an existing gene and mutating that gene and changing it in some way, or taking an existing gene and duplicating that gene and then mutating the duplicate gene, or doing gene regulatory changes and other types of what I would call intrinsic changes in the organism. And I've always peripherally looked at this other topic, but I've basically turned it into everything we work on, which is how organisms acquire functions from the outside rather than from the inside. And that's largely through symbioses, so with interactions with other organisms, or stealing DNA, something called lateral gene transfer. And so almost everything that I work on now is how either a host interacts with a microbial community to allow the host to carry out some new function, or how a community of microbes interact with each other to allow the community to do something that individual microbes were unable to do.
You mean non-Mendelian gene transfer?
Yes, we will talk about that later in the course. That's one of the whole days on genomics. That's the sort of the scientific topic that I'm interested in. And the way I study this is largely through grinding stuff up and sequencing it, and reading DNA sequence data and doing bioinformatics. That's what most of what my lab does. So there are a lot of different ways that you could study symbioses and interactions among community members. The way I do it is leveraging DNA sequence information. And so I'm a little bit different than what the way many other people do their research, in that what I'm interested in is how I take a topic, let's just say photosynthetic symbioses, where one organism is photosynthetic and it helps out another organism by interacting with that other organism. And rather than studying one system and how that works, I'm interested in how all the different versions of photosynthetic symbioses work and what that tells us about the rules by which these types of interactions occur. So you can generally call this convergent evolution. So I would, if I was studying biomechanics of swimming, I would be the person studying sharks and dolphins and the larval invertebrates swimming in the oceans and so on, rather than taking sharks and studying them in a lot of detail. So we change the biological system that we're studying pretty often in my lab, but that's because I want to cover the spectrum of all the different ways that an organism acquires carbon through a symbiont, for example. So we're all over the place right now. We focus mostly on 4 or 5 ecosystems, but that will change, in 2 years or something to that effect. So what I want to do now is just give a little bit of an introduction and go through those 4 eras to give you the perspective before we start talking about each of the different each of the different topics. And so I'm presuming that most people know this, but I will go through it again. I'm just going to talk a tiny bit about an introduction to microbial diversity. And so we're going to focus primarily on microorganisms in this course. What we're learning about applies to other types of organisms as well. So when we talk about genome sequencing, the general approach could work with plants and animals and other multicellular organisms, but we're going to focus on microbes largely because that's what I know. That's where most of the work has been done, actually, in most of these areas. So microbes are defined as organisms that you can't see without the aid of a microscope. Now, it turns out that that definition is really lame. It makes for all sorts of confusing issues because there are animals that are tiny, incredibly, incredibly small nematodes that have, 200 cells in their body. And you can't see them without the aid of a microscope. And there are bacteria that are gargantuan, that are the size of, a pinhead, much bigger than those nematodes. And there are single-celled eukaryotic organisms that are the size of your fist. So, the defining organisms based upon their size turns out to be a really awkward way to classify the world around you. And so when we talk about microbes, we're actually going to focus, and I'll get back to this in a minute, on evolutionary groups of organisms as opposed to their size. And basically, you could view this, and if you've taken BIS 2C, this is basically everything except plants, animals, and fungi. That's the rest of the world is what we're sort of calling microorganisms. Now, there are some exceptions to that, but let's just sort of generally leave it there. We're going to cover a lot of diversity here. The diversity that we see, and there are lots of different aspects of that diversity, which I'll get to in one second, is enormous. The amount of diversity that you see in microbes is very, very large. And perhaps the most important thing for a lot of this course is that the appearance of a microbe, the physical appearance when you actually look at it in a microscope, if they are that small, is not generally a good indicator of either what type it is, that is evolutionarily what group of organisms it comes from, or what it's capable of doing, what its functions are. And this is pretty different than plants and animals and multicellular fungi. I mean, if you go to a new island and you show up at that island and you see things flying around, see something with wings, even if you don't see it flying, you can guess pretty much that it probably is able to fly. And if you see something that is green, you can guess pretty much that it's doing something with certain wavelengths of light, although, not always. And you can learn a lot from plants and animals by just looking at their physical appearance. And their physical appearance is also a very good, although not perfect, indicator of what kind of organism they are. So, just this morning I was going bird watching. I have a bird field guide. The groups of birds that are all in one order or one, family, they generally look similar to each other. And they look different from the birds in other groups. Now, occasionally you have convergent evolution where unrelated birds or distantly related birds look very similar to each other. We have that just in spades in microbes. Very closely related microbes can look very different from each other. Very distantly related microbes can look very similar to each other. Their appearance basically evolves incredibly rapidly, and there's just very few features in many of them, especially like bacteria, single-celled, very small organisms. They don't have a lot of morphological features that you can use to classify them. What this means is that if you wanted to go out to the field and figure out what microbes were in a field sample or in a human gut sample or a skin sample, if you looked at them in the microscope, you wouldn't learn a ton about what was in that sample. And that's very different, again, than what we see for plants and animals. And we'll come back to this in the rest of the course. Now, even though I just told you that appearance is not a great indicator of what kind of microbe is in a sample or what they are doing in a sample, this does not mean that there isn't a diversity of phenotype. among microbes. There is actually quite a bit of diversity. There are some groups like diatoms, for example, that we will talk about, where the morphology is actually an incredibly good indicator of the type of organism that they are. But generally, across all microbes, morphology is not very informative. But you see incredible diversity. So this is spirochete, a spiral-shaped bacteria. This is an archaea, one of the other groups of microorganisms with these two symbiotic archaea that live in association. These are two different species. This is an amoeba. This is one of these giant single-celled eukaryotes, lives in the bottom of the ocean, the size of your fist, leaves trails on the bottom of the ocean in the mud. A lot of microbial eukaryotes have incredibly diverse forms that are largely determined by extracellular silica and other materials that they secrete. There's a very strange salt-loving archaea that is square-shaped. Nobody has any idea, I think, to this day, how it's able to make cells that are square-shaped. And anyway, there's just incredible diversity, and there are many things in the evolutionary groups of bacteria and other mostly single-celled eukaryotes that also do form multicellular structures. And like I was saying, here's this one bacterium. I think it's the one that actually Doug Nelson here in microbiology discovered, this giant sulfur-utilizing bacteria that's, you know, the size of the head of a bee. So there is incredible diversity in the form. And it's not that it is not useful for a variety of purposes, but it's on average less informative than it is for other organisms. There's also incredible diversity of function. And we'll talk about this again throughout the course. You know, most of what people studied for 200 years about microbes or even more were the ones that made us sick. And there's still most of the attention is on the ones that make us sick. And there are plenty of them. They are very interesting. And they're the causative agents of, you know, a huge number of human, plant, and animal diseases, some of which aren't a big deal anymore and some of which still are. There are lots of examples. This is more what I work on in my lab of beneficial microbes that live in association with some other organism and help them out in some way. An example of this are the nitrogen-fixing microbes that live in association of the roots of legumes. So beans and all plants that are in the legume family, they're able to grow in nitrogen-poor soils. And in fact, many farmers use them to pull nitrogen out of the air, and then they just plow them over into their soil because they have an association with these bacteria that are able to take atmospheric nitrogen and turn it into nitrogen materials that are able to be utilized by the plants. There are also an incredible diversity of what are called extremophiles among microorganisms. So if you take a particular environment where you basically look at the range of conditions at which organisms are able to live and really thrive in that particular environmental condition. It could be temperature, pH, radiation levels, pressure, anything like that. At the tail ends of that distribution, you usually find microorganisms are the ones that are able to survive the most extreme conditions of any particular environment on the planet. Next to the pathogens, we probably know the most about microbes that we use for various, usually fermentation processes or other types of agricultural and food production processes. So, you know, Saccharomyces cerevisiae, which is used for beer, wine, bread, was the first eukaryote to have its genome sequenced, and we know an incredible amount about it. There's a lot of work expanding still, but probably not as much as it was 5 or 6 years ago, on using microbes to produce things like biofuels. And microbes play fundamental roles in most, if not all, of the global nutrient cycles on the planet, and therefore have impact on all sorts of environmental processes. So for the course, we're going to talk about all of these different aspects of diversity, and for many parts of the course, we're going to orient ourselves with evolutionary diversity. That turns out to be one of the most useful ways to deal with the great diversity of any type of organism. And so if you, as we will talk about later in the course, if you build an evolutionary tree of organisms, you can classify organisms into different groups based upon that tree. We'll talk in more detail about this. And I'm not going to go into a lot of detail unless we get to it at the end of today about how to read and interpret a phylogenetic tree. You should know this. If you don't, I can send anybody review papers and other things. But we will see a lot of evolutionary trees throughout the course because that turns out to be one of the most powerful tools in analyzing all the sequence data that people have been generating. So many of the papers we will be reading, there will be some evolutionary tree as part of the paper. They may not care about evolution, but they use evolutionary trees to figure out something about the organisms that are being studied. It's not particularly novel. That is, of course, how we deal with plant and animal diversity too. So again, if you need a refresher, I'm happy to send people refreshers because we really will spend a lot of time looking at phylogenetic trees, but I'm assuming that you know how to look at a phylogenetic tree, what they generally mean, and a little bit about how they're made, although we're not going to talk in a lot of detail about how they're made. Okay, so are there any questions about that so far before we get into sort of the overview of these 4 eras? All right, so I've sort of, there's a paper, it's linked on the SMART site. I posted, oh, I didn't say this. I posted under syllabus and SMART site an outline of the course with the readings for each week, but I will, I'll send around an email announcement to everybody about that too. There is a reading for the next lecture that is posted there. It's a review paper on DNA sequencing methods that you should read before the next class. This is a paper that sort of covers the introduction that I'm about to do. At least it's a very general summary of these 4 eras of DNA sequencing and microbial diversity. And again, what we're going to do, and I'm sort of going to give you a perspective on the whole course here. So for the next class on Thursday, we're going to talk about DNA sequencing, the history of it, and sort of the crazy revolutions that are going on now. And mostly what people talk about with DNA sequencing is how the cost and sort of the equivalent, the ease of doing DNA sequencing has improved at a rate faster than this classic presentation, which is known as Moore's Law, which Gordon Moore from Intel basically came up with this law that predicted that the cost of computing or the cost of disk storage or the cost of CPU processor time would decrease exponentially over time. And the cost of sequencing has decreased even more than that over time. And this is why DNA sequencing is now used for all sorts of purposes that it wasn't even used 5 years ago. It's why genomics is still being talked about, even though some of us thought it was dead a few years ago. And this basically means that if there's some information that you can extract out of the DNA of an organism or even an artificial piece of DNA, you can now read that information at an incredibly low cost. And I'll just give you an example of this. So the way sequencing used to be done, and we'll talk about this on Thursday, you would generate, in essence, sequence information for a single piece of DNA that came from your organism of interest at one time, run one sequencing reaction. I did this as an undergraduate, and for the year after I graduated, I worked in a lab as a technician. This was in 1989 and 1990. I sequenced a single gene over a year and a half, 1,400 base pairs. It probably cost about $50,000 in reagents and other costs. And we got 1,500 base pairs of DNA sequence data out of the organism of interest. And I got a paper out of this. 10 years ago, when the Human Genome Project was just being finished, we thought that there had been this amazing revolution in DNA sequencing whereby it cost, I worked at a big genome center before moving to UCDavis that sort of generated large amounts of genome data. It cost about 50 cents to read 1,000 bases of DNA at that time. And we were just like, oh, we got to sequence everything. It's amazing. Oh, we got to sequence the planet. And today, in a lab in the Genome Center here at Davis, we can generate about 100 million bases of sequence for $1,000. So that means that if there's some information in the DNA sequence of an organism, we can generate a massive amount of it for basically for less than it costs to pay the person to watch the machine to generate the data. Whereas previously, if you got a grant to sequence the genome of an organism, that was $1 million grant. Now we can sequence the genome of a bacterium for $50. So it's just changed what you can imagine doing, such that in the clinic here in the hospital, if you get an infection in the hospital and it doesn't respond to antibiotics, they may actually sequence the genome of that organism to try and figure out what antibiotics to use. And that was unheard of 4 years ago. And that's what we'll talk about in the next lecture. And now what I'm going to do is give you just sort of this overview of these 4 eras. And again, these eras started with this, the tree of life, and each one of these eras continues after it started, so they're not getting rid of the other eras. So basically, DNA sequencing or reading the sequence information from the genome of organisms started, probably in the early 1970s, when people were really first trying to do this. And it was painful, it was very slow and very expensive, but eventually the people who developed the best methods to do this, Wally Gilbert and Frederick Sanger, 2 different methods for reading sequence information. They won the Nobel Prize for this, even though, compared to today, it's kind of like a joke what the people were doing with DNA sequencing. But most of what people were doing with DNA sequencing involved things like trying to type sickle cell anemia loci within different humans to try and understand what the mutations were that people had that led to particular disease phenotypes. And there was obviously, as always, a lot of focus on humans. And what we're going to do in lectures 3 and 4 is talk about sort of the history and then the modern view of using DNA sequencing to build an evolutionary tree of organisms, the so-called tree of life. And there have been attempts to build a tree of life for many, many years, ever since Darwin basically convinced people or convinced a reasonable number of scientists that all organisms on the planet were related to each other by some type of evolutionary tree. People started to try and figure out how can we draw a tree that includes as many organisms as possible. It was pretty straightforward for people, even before Darwin, but certainly after Darwin, to say, let's draw a tree of mammals and try and figure out, here are the the ungulates, the cows and the horses and the camels, and they all seem to group together, and we could probably draw a tree for them. And eventually there were sort of models that were developed on how to build that tree. But how could you connect together plants 150 years ago with animals when nobody had ever looked inside cells of any organism before? And how could you connect these to microorganisms, which had no obvious similarities in any feature that you could actually measure to other organisms? And what people largely did, like this very famous person, Ernst Haeckel, was, let's be polite, but was just make shit up. They came up with thought models. They were very important. They came up with basically thought models for how organisms should have evolved based upon what they look like today. But there was virtually no data behind these trees that people drew. So Haeckel came up with this tree, which is, reasonable, but it's largely based upon, again, just sort of a thought experiment that had one major branch with what he called animalia, that's basically what we call animals today, one large branch with plants, and one large branch with what he called protista. These were basically the single-celled eukaryotes, the things that looked, when you looked in a microscope, like the cells of animals and plants. I'm not 100% sure if they knew what a nucleus was back then, but phenotypically they were complicated and diverse, and they looked like the cells of plants and animals or some way. And then down at the bottom, he just dumped, basically the bacteria down at the bottom of the tree, and he's like, I have no friggin' idea what to do about this. I can't even come up with a model. And then there were many other concepts for this. In particular, one that is still in many textbooks today is this one from Whitaker, Five Kingdom Tree, where he said, actually, we should really add, basically he said, those three aren't enough. Let's add fungi in as their own group. Let's put protists in this really weird spot at the bottom of the tree where I just, going to draw lots of little branches pointing to different things, and I have no idea what to do with these single-cell eukaryotes. And then bacteria, which he now had at least a few different branches of, he still had no real idea what to do with, and put them sort of at the bottom of the tree. And again, this was not for the bacteria part of this tree and for the protists. There wasn't data behind a lot of this. This was, again, thought models in many cases about the tree of life. And that changed basically with this one person, Carl Woese, in the 1970s. And what he was doing was he wasn't really interested in the tree of life itself, per se. What he was actually doing was studying the mechanisms by which organisms translated RNA into protein. So he was a cellular molecular biologist trying to figure out the rules of the genetic code. This was the era when the central dogma of molecular biology was being determined. People sort of figured out that DNA was the genetic material, DNA went to RNA, RNA went to protein, that's the central dogma, but they didn't know at the time how RNA was translated into protein. And so there were a lot of people at the time that were taking cells of different organisms, breaking them apart, and isolating the machines in those organisms, the ribosomes, that seemed to be where RNA was being translated into protein. And some people were trying to figure out what the actual code was. There were lots of interesting experiments related to trying to figure out what whether it was a 2-letter code or a 3-letter code or a 4-letter code. And Woese was one of the people doing some of those experiments. But for reasons I'm not actually 100% sure of, one of the things that he started to do was to take out the ribosomes from cells. So you crack open a cell, the cell is filled with all sorts of things, and you could physically isolate the ribosomes, the machines that are translating RNA into protein, using centrifugation. You spin the cells after you crack them open with these really high-speed centrifuges, and different parts of the cell have different speeds at which they will flow through different materials that you mix them with. So you can mix them with like some sort of matrix, for example, and large things would go slower through the matrix than small things. You could also spin them with various other chemicals such that the density of a molecule would determine where it showed up in these centrifugations. And what Woese and other people developed was ways to physically isolate out the different components. So the ribosome, this big machine, it's a tiny little machine inside cells, but it's big compared to the rest of the stuff in the cell, is made up of actually two major different subunits, the large subunit and the small subunit. And those are made up of different subcomponents. Those subunits are made up of a group of proteins, the large subunit proteins and the small subunit proteins, and each of them has an RNA molecule inside of them as well. And most people were interested in the proteins inside the ribosome. There was a theory that all cellular processes were basically carried out by proteins, and therefore RNA was uninteresting. And Woese had written a bunch of papers at the time about hypothesizing about the origin of life and how it made sense that RNA was the original molecule for heredity during the origin of life on the planet. And the reason for this was that RNA is much more chemically active than DNA. RNA can react with more things, and he and a few people had this model that would allow you to have heritable material that was also enzymatic. So we got really interested in the RNA component of the ribosome when most people couldn't care less about the RNA component. And he took a method that a few other people had developed that allowed you to take the RNA component of the ribosome. So there are 3 major RNA components of the ribosome. And if you do this in bacteria, they got called 5S, 16S, and 23S. S stands for sedimentation. When you did this centrifugation with certain conditions, this was the biggest one, the second biggest, and the smallest one component. These are now RNA. So a cell has a genome. It transcribes its genome to make the RNA of the cell. Most of that RNA is then translated into protein. But some of that RNA is kept as RNA, tRNAs that function in translation of the messenger RNA, and what are called ribosomal RNAs show up in the ribosome. They never get translated. They function as RNA.
So they're considered to be structural RNAs then?
You could say that, but a few years ago, Harry Noller showed that the catalytic component of the ribosome is actually these things. So they're not, they actually are the thing that carries out the protein synthesis reaction in the ribosome. So they thought at the time that these were structural components. They're actually the enzymatic component of the ribosome.
So you have more than a few groups of, differentiations of RNAs then?
You have these 3 in the ribosomes. So cells, you make tRNA, ribosomal RNA, and messenger RNA from the genome of a cell. These are what get made into proteins. So in a bacterium, E. coli has 5,000 or so genes. 4,950 of them are for proteins. And a small number of them are for tRNAs and RNAs. We'll get back to this when we talk about genomics of organisms. But every organism on the planet has ribosomes. Those are the machines in every organism that carry out this translation of RNA into protein. And again, Woese was really interested in these partly because of this idea that early in evolution, RNA might have been this important part of the cell. And so he would take a test tube filled with some organism that was grown in the lab, or you'd take tissue from some plant or animal, run it in one of these centrifuges where he could physically separate out the 5S, the 16S, and the 23S. And then what he did was, for these different things, he could then chop them up into smaller pieces and run a special method that separated those pieces from each other. It's called 2-dimensional electrophoresis. And you end up with these little spots. And then he would physically cut out of this 2-dimensional piece of paper, basically, those spots, and run a chemical reaction that could read 8 letters of their sequence. So we took this for E. coli. He did these 13 organisms here: Saccharomyces cerevisiae, 1 eukaryote, Lemna minor, I think that's a plant, although I'm not sure, and then a animal cell line. A bunch of different bacteria and a bunch of different other organisms. And he ran this reaction, and then he cut out these little pieces of the ribosome, and he read the letters that made up those pieces of the ribosome. Tiny little bit of the sequence of the RNA from these organisms. You can infer from what the RNA is what the DNA should be, because you know that DNA gets made into RNA by the complementary rules of DNA base pairing. And what he did then, cumbersome though it may be today, he made a catalog. And he said, organism 1, one of the little pieces looked like this. Actually, it's RNA. RNA has U instead of T. And then maybe a second piece looked like U, C, C, G, G, A. And then he made a table of all of these 8 base pair pieces of RNA. And he said, organism 1, does it have this? Organism 2, does it have this? And he just made this big table of which organisms had these tiny little 8 base pieces of their ribosome. Because when he did this, he didn't know where in the ribosome this little piece came from. He just knew that this organism, when you cut out the piece and you read a little bit of the string of letters, somewhere in that organism's ribosome there was an AAAUCAGC. And somewhere in organism 2 there was that same string of letters. They may not have come from the same place, but he didn't know any. This is all he knew. Made this table, and then he compared organisms to each other, and based upon how many of these little, they're called oligomers, how many they shared with each other, he just gave them a similarity score. These two organisms, if they shared all of them, they would have a 100% similarity score. And he called this association coefficients. And we're going to read his paper and talk about this next week. And he took those organisms and he grouped them based upon their similarity score. And he found this striking thing. We're going to get back to this, so I'm not going to go into a lot of detail here. But basically, he had taken organisms, all of these organisms, 4 through 13, were classified as prokaryotes. They were grouped together into the same group of organisms because they did not have a nucleus. And they were all thought to be basically equivalent to each other. And they should all be more similar to each other than any of them were to eukaryotes. And instead, what he found was 3 groups of organisms instead of 2. The prokaryotes were split into these 2 groups, where within the group, everybody was more similar to each other, and between the groups, they weren't that similar to each other. And this was basically the discovery of what's now called the archaea. It's the 3rd branch on the tree of life, and it was hidden within what had been called prokaryotes before Woese did this ribosomal sequencing. And this reading of DNA sequence data and then trying to group organisms based upon sort of mathematical analysis of data rather than thought experiments was the first time that people had actually tried to build a tree of life representing all organisms using objective, data-driven methods. And what he discovered was this hidden group of organisms, what he originally called archaebacteria. They were then renamed the archaea, sort of hidden among the other organisms that had been called prokaryotes. And a few years later, that we will get to when we talk about it, he actually built an evolutionary tree for these organisms and showed this pattern, where here you have eukaryotes, the organisms with nuclei. Here you have the regular bacteria that people were used to dealing with. All of the bacteria that cause diseases are in here. And then this 3rd group, again, which he called the archaebacteria and now known as archaea. So this was the era 1, was taking organisms that you could study in the lab and generating DNA sequence data for them, or in Woese's case, RNA sequence data, but it's equivalent, and then comparing organisms to each other and inferring how they're related to each other based upon their sequence data. And this primarily for many years focused on doing this by looking at RNA, ribosomal RNA genes. And by doing this, the classification of organisms on the planet was completely rewritten. And in many sort of esoteric ways, but many fundamentally important ways. So people basically went through, once Woese, some people didn't believe this, but this was on, I think, the front page of the New York Times when he published this paper. I mean, this was a big deal in the scientific community. In 1977, we're going to read the 2 papers that were published on this. That's the history of the field. And immediately, everybody who worked on any prokaryote started to sequence the ribosomal RNA genes from that organism and try and figure out what it was related to. And it led to massive changes, not just in sort of concepts, but in medical treatments for various diseases. For example, prior to this work, rickettsias, which include the organisms that cause things like Rocky Mountain spotted fever and a variety of other tick-borne diseases, and chlamydias, which cause things like chlamydia and trachoma, causative agent of blindness throughout developing countries, were grouped together. And in addition, many people classified them as viruses. Turns out they are bacteria. Viruses don't have cells. These were very small organisms, so they were misclassified as viruses because many people thought they were just too small to be cells. And when people did ribosomal RNA sequencing, they discovered that they were not even remotely closely related. And people had been using like one class of antibiotics to treat chlamydial and rickettsial infections because they thought they were in the same group of organisms. Turns out rickettsias are actually in the same broad group as E. coli, and chlamydias are in their own major separate lineage, and they don't respond to the same type of treatments, not surprisingly, because they've been separated by billions of years of evolution. And so you see, if you go back and look at the literature, which I don't really recommend doing all of, every 3 months there was a new paper saying, oh my God, we called this thing X, and really it's Y. So for example, there's things that used to be called Pseudomonas. Many people may be familiar with some Pseudomonas species, like one of the infections that kills a lot of people with cystic fibrosis is caused by a bacteria in the Pseudomonas group. Now that people did ribosomal RNA sequencing, the organisms that were called Pseudomonas actually come from something like 35 different groups of organisms. I mean, they're just all over the place, and they were all lumped together into Pseudomonas because they all had some similar phenotypic property that was the screen used to call things Pseudomonas. And many of them don't respond to the same antibiotics. They don't respond to the same type of treatments. Anyway, so this led to what you could really call the first phase, the first era of sequencing and microbial diversity to try and figure out what organisms were based upon their ribosomal RNA sequence. And this was done largely for organisms that you could grow in the lab. And the main reason for this was that if you come back to this, when Woese was doing this originally, you needed large amounts of cells of organisms to physically isolate large amounts of these ribosomes from the cells. That would eventually give you enough material to run this two-dimensional gel, or eventually they got rid of that and ran some other methods. But you needed gobs and gobs of material, and it needed to be really pure and really clean to get this to work well. And this only worked really well when you had an organism growing in the lab in a clean environment, or you could take a tissue from like a plant, but it had to be one organism at a time. And this limited what people were doing to figure out what types of organisms were on the planet to things that you could grow in the laboratory. And we'll come back to all this tree of life stuff. As just an aside, one of the papers that we will talk about when we talk about the tree of life is now saying that the 3 lineages that Woese claimed existed based upon ribosomal RNA analysis might in fact be slightly wrong. And that if you build an evolutionary tree of everything now that people have characterized, it looks like the things that were called archaea are actually many different lineages, and eukaryotes may be sort of a separate sub-branch of archaea, but we'll come back to that. There's some fine tuning that needs to be done compared to what Woese did. So, but the next era was saying, okay, so we can grow organisms in the lab, but what about everything else on the planet? And that's what we're going to talk about in lectures 5 through 9, is how we can apply what Woese invented basically to studying organisms that we can't grow in the laboratory. And the reason for doing this is that for plants and animals, you can do lots of field studies to learn about the organisms in the environment. You can go collect them. You can do all sorts of controlled ecological experiments. You can even use satellite imagery and do all sorts of cool things with plant and animal diversity. You can do this with microbes. There's lots of great microbial fieldwork that is done everywhere in the world and is even done here. There's people who go out to hot springs and collect samples and study the microbes there. There are people that do controlled ecological transects. We can use satellite imagery to look at chlorophyll absorbance. We can go out with a microscope in the field and do lots of things. But again, because the appearance of a microbe is not a good indicator of who they are or what they're doing, it's limited. The field work that we can do has a couple of major limitations. And what people, and this is a big problem, because the main way that people were getting material for this type of experiment or for any type of experiment on microbes was to grow them in isolation in the laboratory. So you take a sample, you go through many steps until you eventually get a single cell from the organism of interest, and then you clone it, basically grow it in the lab where it reproduces asexually, and you get lots of copies of an individual cell, and then you can study its biochemistry, or its genetics, or its physiology. And this is a big problem because most organisms on the planet can't be grown in the lab. So if you go to any ecosystem, like a Yellowstone hot spring or anywhere, and you take a sample and you try and grow everything in that sample, and you take another half of the sample and you look at it in the microscope and you count the cells that you can observe in the sample, you always see many more cells in the microscope than you are able to grow things from. It's called the great plate count anomaly because they would count the number of cells that you could grow by putting things onto agar plates. And this is very vexing if you want to learn about microbial diversity. And if most of the microbes on the planet can't be grown in the lab, and the only way that we're really learning about them is growing them in the lab, this was a big problem. And one of the solutions to this problem came in the 1980s, where someone took Wose's approach and said, why can't we do this for organisms from the environment? It's a little trickier, but let's try and do it. So let's go to an environmental sample and try and pull out, originally when they first did this, and we'll read some of these papers, they actually originally ground up environmental samples, extracted the ribosomes from those samples, and then read some of the sequence information from these mixed samples that contain multiple organisms at one time, and then tried to figure out what organisms were in those mixed samples by doing a similar analysis to what Wose did, but now a little more complicated because there are many organisms in the sample. This was revolutionized. So this was originally pioneered by a guy named Norm Pace and his colleagues. And then this was completely revolutionized in originally in 1985, but really in 1988 when this method called PCR, the polymerase chain reaction, first became commercially available. And what PCR allows you to do is take, if you know for an organism of interest, parts of the genome of the organism of interest that are close to each other in the genome, you can make what are called primers to run this PCR reaction, and you can make billions of copies of that part of the genome of those organisms using this PCR reaction. And this was invented by someone at a company, and it was largely used for doing things like typing HIV genomes or doing human genetic typing. And the people who worked on ribosomal RNA, environmental sequencing, heard about this PCR reaction, and they said, oh my God, that's amazing. Because there's this really cool thing about ribosomal RNA in organisms. The reason that Carl Wose was able to tell the difference between organisms when he built his tree is that organisms differ in the string of letters inside their ribosomal RNA. Closely related organisms are more similar to each other than distantly related organisms, just like closely related vertebrates have similar bone structures. Same thing with sequence information. But the great thing about ribosomal RNA, which was basically set up for PCR, is that across all organisms on the planet, there are parts of these ribosomal RNA genes that are nearly identical in everything. They're the parts that carry out this catalytic reaction of making proteins. And they're basically identical in sequence across all organisms. And what this means is that even though PCR was invented largely to get one region of a single organism's genome out at a time, in one reaction, you can make copies of all of the ribosomal RNA genes from all of the microbes in the sample at once using this PCR reaction. So you add these PCR primers, and if there's one organism in your sample, you get out a copy, billions of copies of its ribosomal RNA genes. And if there are 2 organisms in the sample, you get out billions of copies of each of those 2 organisms' ribosomal RNA genes. And if you run sequencing now on those samples, you can read the sequence of organism 1 and the sequence of organism 2. And if there are 4 organisms in the sample, you can get out the sequence of 4 organisms' ribosomal RNA genes. And now you've never grown the organism, but you can tell what kind of organism was in the sample by building an evolutionary tree of the ribosomal RNA genes and looking at how it compares to known organisms. It's sort of the equivalent of what paleontologists do with a tiny little bone fragment. Go to a paleontology site, you find a tiny little piece of a femur. But if you have a good database of femurs from other organisms, you can figure out that that tiny little piece tells me that a tyrannosaur was in this sample. We do the same thing with ribosomal RNA for microbes. And if there's 1,000 organisms, you can do the same thing if DNA sequencing gets cheap. And so for that second era, we're going to talk about what you can learn about diversity of microbes by doing ribosomal RNA sequencing as a surrogate for learning about what organisms are present in a particular sample. And there's lots that we can learn. We will do a case study on the human microbiome and a case study on the building microbiome. And then we will talk in era 3 is going back to growing organisms in the lab and saying, ribosomal RNA gene, whoop-de-doo, that tells me, I can build an evolutionary tree, but that doesn't tell me much else about the organism. And in 1995, the first complete genome sequence of any organism was determined, this bacterium Haemophilus influenza. And that launched sort of the genome sequencing revolution, and there's been incredibly powerful use of genome sequencing to characterize microorganisms as well as other organisms. It allows you to do things like predict what their processes are, do better vaccine and drug design, learn about their evolution. Having the genome serves as a template for functional studies, and so on. So we will spend 4 or 5, I can't remember off the top of my head, classes on talking about how you sequence a genome, the history of genome sequencing studies, and what you can do with genome sequence data, what it tells us about microbial diversity, function, and evolution. And we're going to do a case study of a project that actually I coordinated on trying to sequence genomes from across the diversity of life. And then for the last part of the course, we're going to talk about equivalent to with ribosomal RNA, saying, okay, we went to the environment, we sequenced these ribosomal RNA genes, they told us what organisms were in the environment. That's not enough. We want to learn about the functions of organisms in the environment. We want to learn more than just the ribosomal RNA. Let's go back and say, rather than running PCR reactions from these environmental samples, can we sequence the genomes of organisms without ever growing them in the lab? And that's what's generally called metagenomics. We're going to talk about the history of the field. There's an incredibly cool series of papers in history involved, which launched this field by someone named Ed DeLong, who was at the Monterey Bay Aquarium Research Institute. And we'll talk a little bit about what he found with their original studies of metagenomes. And then we're going to talk, they were doing sort of a painstakingly slow process. And then in 2004, 2 papers came out where people applied sort of random, more big-scale methods to sequencing genomes from environmental samples. We will talk about those, and we're also going to talk a little bit about how hard it is to analyze this type of data and what you can do, even though it's a little bit messy, with learning about microbial, functional, and ecological diversity from this metagenomic data. And we'll do another case study talking about the human microbiome and what people have started to do with reading genome sequence information from microbes in our gut or on our skin or other places on the human body, and what you can learn about things like health and disease states from analyzing that data. And then that's it. So that's basically the 4 eras: tree of life with ribosomal RNA, ribosomal RNA from environmental samples, genomes from the lab, genomes from environmental samples. And in each case, we will do the history, we will do what we know now, and we will do a case study. And at some point, we will figure out a way for you to select 1 or 2 papers to use as your course topics in these areas.